Sexual reproduction in flowering plants. Theory board exam preparation 2025. First question. Write a neat labeled diagram. Describe the structure of dithecous anther. It will be asked for 5 marks. Here is a neat labeled diagram of a dithecous anther with labelings: connective epidermis, endothelium, sporogenous tissue, and tapetum. A typical microsporangium has 4 walls. The first one is epidermis, it is the outermost single layer. Then comes endothelium, it is the second layer. And middle layer, it is 1 to 3 layers of a cell, and it is protective in nature. The fourth one, that is tapetum, it is the innermost and it provides nourishment to developing pollen mother cells. And each microsporangium contain a sporogenous cells and produce pollen grains by meiosis. Second one, write a neat labeled diagram and describe structure of pollen grain or male gametophyte. It will be asked for 3 or 5 marks. Exine, it is a outermost wall, whereas intine, it is a innermost wall. Then intine is continuous layer, whereas exine has a small pore, that is germ pore. And we will find 2 different type of cells. One is vegetative cell, another one is generative cell. The pollen grain or microscope represent male gametophyte, and it has 2 layers, exine and intine. Exine is made up of a material, that is sporopollenin. And because of the presence of sporopollenin, pollen can be stored as a fossil. And this one is very important regarding exine. So exine is absent, that will be called as germ pore. And intine, it is made up of cellulose and pectin. It is also important question. And pollen grain has 2 cells, vegetative and generative cell. Vegetative cell is large and abundant rhizophore, whereas generative cell is small. So it is diagram with explanation. Third one, write a neat labeled diagram. Describe the structure of anatropous ovule. So they may ask, draw a neat labeled diagram of megasporangium. You have to draw the same diagram. Megasporangium. So here are the labelings. So these are integuments, outer and inner integument. They play a role in protection. Funicle, it is a stalk of the ovule. Hilum, it is a junction between the ovule and the funicle. Then integuments, ovule is surrounded by 1 or 2 protective layers. Then micropyle is a small opening present at the tip where the integuments are absent. It is not completely enclosing. So then micropiles, small opening aitu. Chalaja, it is a ovule opposite to micropyle. Nucellus, it is a rhizofood material. Then embryo sac is a female gametophyte located within the nucellus. That is female gametophyte. It is very important for KCT and NEET, and this particular diagram will be asked as a match the following regarding hilum, funicle, micropyle and chalajal pole. Then fourth question, what is megasporogenesis? Explain the development of 8 nucleate female gametophyte. 5 mark question. Here megaspore mother cell, it will undergo division to form a megaspore dyad. that is 2 megaspores will get formed. Again it will undergo second division to form a megaspore tetrad. 4 different megaspores will get formed, where in these 4, 3 will get degenerated, only 1 will be considered as a functional megaspore. So this one functional megaspore develops into female gametophyte. Female gametophyte, that is embryo sac. So megasporogenesis, it is a formation of megaspores from the megaspore mother cell, that is MMC. Development of female gametophyte explained with the neat labeled diagram. So functional megaspore, it will divide mitotically to form a particular mature embryo sac. So it will undergo 2 mitotic divisions and 8. So it will undergo continuous mitotic divisions and it will form 8 different cells, and these cells, they will get surrounded by a wall. Then they will get formed as 7-celled 8-nucleated embryo sac. So 3 cells are antipodals, 2 synergids, 1 egg cell and 1 central cell. So total 7 cells. The female gametophyte is 7-celled and 8-nucleate structure. Megaspore tetrad, which results into a formation of a functional megaspore. It will undergo mitotic divisions. It forms a 4-nucleate and 8-nucleate stage. After that 8-nucleate stage, the cell wall get formed. In egg apparatus, you will come across 2 synergids and 1 egg cell. Whereas at the chalaja, you will find 3 antipodal cells. Next question. Write a neat label diagram, describe a structure of mature embryo sac. So embryo sac, it contains a egg apparatus at micropylar end, 2 synergids and 1 egg, whereas filiform appendages will be guiding a pollen tube towards the egg, and antipodals, 3 antipodal cells at chalaja, 1 at central cell. So totally embryo sac is a 7-celled and 8-nucleated. Chalaja, it will be remembered as CA. Chalaja, antipodals, micropylar and MES, that is egg and synergids. Centrally we have central cell. Within that you will find 2 polar nuclei. So 7-celled, 8-nucleated. Here 3, here 3, 6, at center 2. Next question, what are cleistogamous flowers? Write their significance. Cleisto means they do not open, where anther and stigma, they lie close to each other, and significance, invariably autogamous, no chance for cross pollination, and you will find a assured seed set. Next question, what is pollination? Mention any 4 features of wind pollinated flowers. Pollination, that is transfer of pollen grains from anther to stigma. So in wind pollination, now pollen grains are light and non-sticky, exposed stamens, large feathery stigma, then flowers are small and colorless, nectarless and inconspicuous.